Okay, well, welcome everyone. Thank you for joining us today. My name is Maggie, and I'm the Director of Development and Communications for the EHE Foundation. Before we get started, I just want to share a few housekeeping items. As you came in, it probably gave you the notification that we are recording this. So if you find you need to leave before it's over, no problem. We will be posting the recording to our website and YouTube channel soon, next week, mostly. I want to call your attention to the Zoom toolbar down below at the bottom of the screen. The most important thing down there is the Q&A function. We encourage you to ask questions. That's where you want to put them, not in the chat. Please put them in the Q&A. That's where our team is going to be looking to find the questions and kind of triage them to Dr. Lamar. So please use the Q&A for any questions you have and put them in as you think of them. We'll probably just get to those all at the end for the most part, but go ahead and put them in as you're thinking of them. And then we'd like to remind everyone that the EHE Foundation does not provide medical advice. The information provided here is for your information only, and we encourage you to contact your healthcare provider for any specific concerns or questions about your health. Okay, well, our Executive Director and Director of Research is Denise Robinson, and she's going to go ahead and get us started today.
Hey, everybody. Hi. Good to see you. Thanks for making time on your Saturday to join this session of EHE 360 Connect. I have the pleasure of introducing today's presenter and a true champion for the EHE community, Dr. John Lamar. The Lamar Lab at Albany Medical College studies cancer metastasis and has focused heavily on the Hippo pathway with early work in breast cancer and melanoma, dating back to 2012. Thankfully, we have attracted John's talents and more of his time to investigate EHE. He is an active collaborator and a recognized contributor to the advancements in our understanding of EHE. And today you're going to learn more about his current research, all of which is made possible by the generosity of this community through foundation grants. Gratefully, John also lends his time now to the EHE Foundation as a member of our Scientific and Medical Advisory Board. In his spare time, of which I think there's very little, he is an active dad and a mentor to an amazing team in the Lamar Lab. So as Maggie mentioned, put your questions in the Q&A, and then once John's done, we'll go through those questions and talk through those with him. So I'll turn it over to you. Welcome, John.
Great. Thank you, Denise and Maggie, for the nice introduction and the opportunity to participate in this webinar. And thanks to everyone for taking time on their Saturday to hear a little bit about my lab's work. So hopefully we can see the screen.
Yep, we can see you.
All right, good.
Perfect.
So as Denise said, my lab is traditionally been focused on the Hippo pathway, and we've over the last couple of years kind of used that knowledge base to start investigating EHE. And I guess probably for this group, I don't really need to spend a lot of time in this slide, but normally when I'm presenting this, I have to tell everyone what EHE is because they've never heard of it. So it feels a little unusual to not go through the slide. So I will go through this quickly. So EHE is an ultra rare soft tissue sarcoma, which is a type of cancer. We think that the cell of origin, the place that EHE begins, is in endothelial progenitor cells. So these are the cells that make up the blood vessels in the body, and an early stem-like cell of that lineage is where we think the cancer is originating in. Slides aren't advancing. There we go. So EHE has an incidence about 1 in 1 million, which is why it's classified as an ultra-rare cancer. It can affect patients of just about any age, but the median age of onset is about 35 to 40 years. Soft tissue sarcomas, like EHE, can form in just about any soft tissue, but the predominant sites in EHE are the lung, liver, and bone, with the lung, and particularly the pleural EHEs, being the most aggressive and hardest to treat. There are no effective treatments for the rapidly growing aggressive form of EHE, and that's one of the things that we're trying to solve. In addition, EHE has this prolonged indolent course, and about a third of the patients are asymptomatic, often diagnosed incidentally for other reasons. And that those patients can remain in that indolent or stable disease for months, years, even decades in some cases. And because we do not have very good and reliable treatments for EHE, and many of the treatments that are used in the aggressive form of the disease tend to carry side effects with it. A lot of patients with the indolent or stable disease will opt for this active surveillance where they're just monitoring the disease and hoping that it remains in that indolent course. Because it's often presenting as metastatic and diffusely growing disease, so this usually means that you have multiple organs that have EHE in it and multiple lesions in an organ. That makes it very difficult to surgically remove many EHE patients' tumors, and so that's why they tend to opt for this watch and wait or active surveillance. And so the bottom line here is that we need more effective treatments for the aggressive form of EHE, as well as alternative ways to treat or manage this indolent form of EHE besides just active surveillance. It would be great if we could get rid of the disease instead of just watching it and waiting for it to convert to an aggressive form. And so that's kind of the goals that my lab has been focused on. You've probably heard about these two oncogenic fusion proteins that are really the underlying cause of all EHEs. And so about 90% of patients are positive for this gene fusion called WWTR1 campta1, and this encodes the TAS campta1 protein, which I'm going to spend a lot of time talking about. And then the other 10% tend to have this YAPTFE3 gene fusion, which encodes the YAPTFE3 fusion protein. And most of our work focuses on the TAS campta1 fusion because that's the models that we use, but these two fusion genes are very closely related and behave very similarly. So there's a very good chance that a lot of what we're learning in the TAS campta1 driven disease is going to apply to the YAPTFE3 driven disease. And so what is a chromosomal translocation and how does this happen? So you probably know that all the cells in the body have DNA, and that DNA is the blueprints of life. They encode all the things that the cells make, and those individual blueprints, we call them genes. They are organized in these long strands of linear DNA, which the cell then organizes into chromosomes. And every once in a while, when the cell is just kind of doing normal things, these chromosomes can exchange their arms, parts of their arms. And so essentially, a tip of the chromosome, one of the chromosomes will break off and swap places with the tip of another chromosome. And this is kind of what results. And when this happens, if that breaking point where it breaks happens to be in the middle of a gene, you can take part of the blueprint that encodes one protein and attach it to part of the blueprint that encodes another protein, effectively creating a blueprint that encodes a new protein that has a little bit of each of the individual proteins. And that's kind of what happens in the TAS campta1 and the YAPTFE3 fusion. So this is a little cartoon that kind of shows you the different parts of the protein that is normally encoded by the TAS gene, the normal TAS gene. And here's the normal campta gene. And so when this chromosomal translocation happens, the beginning part of the TAS gene gets broken off and attached to the end part of the campta gene. And this is what it looks like in the end. So you have the beginning of TAS, the end of campta1. And when this happens, you lose parts of the TAS protein that allow the cell to control it or regulate it. You also gain some parts of the campta1 protein. And in particular, you gain this very strong part of the protein that forces it to go to the nucleus of the cell. And I'll show you why that's important in a second. And so you've effectively created a hyperactivated or uncontrollable form of the TAS protein. And so the way that this causes cancer is really kind of explained in this Hippo pathway slide. I'm not going to go through the nitty-gritty of this. But this is the pathway that we're talking about. YAP and TAS are two proteins that are very closely related. And what they normally do in cells is they go to the nucleus of the cell when they're active, and they cause the cell to make other proteins. They turn on other genes, and we call that gene expression. And so that they turn on other genes, and these other genes that they turn on tend to promote things like cell proliferation, so the division of cells to make more cells. Cell survival, the cells will last longer than they would normally last without those genes. They make the cells act a little bit more like stem cells. And so this is really important when you want to grow tissues, when you want to get bigger, make organs bigger, or when you want to repair tissues or replace damaged cells. And so it's very important to have this process. However, if you don't control this appropriately and you continue to make more cells and more cells and more cells and not let them die off like they're supposed to, this can eventually lead to cancer and it can make cancer more aggressive. And so the Hippo pathway is a pathway that normally restrains YAP and TAZ. And it does so to prevent this from happening. And so what this pathway does is it senses signals that cells get from other cells in their environment or from the microenvironment itself, other parts of the body, that say, okay, we have enough cells right now. We don't need more cells. So restrain YAP and TAZ, don't let them be active. And that's what the Hippo pathway is supposed to do. In the TAZ-CAMP1 version, in the YAP-TFE3 versions of these genes, you've lost some of the key parts of the protein that allow the Hippo pathway to regulate it. And so now YAP and TAZ are going to the nucleus when they're not supposed to and turning on a bunch of genes that they're not typically supposed to be turning on. And this is leading to the development of cancer. And so in EHE specifically, this is sort of what this looks like. You have this translocation event where the chromosomes exchange parts and you create this TAZ-CAMP1 or YAP-TFE3 fusion. Again, this fusion protein doesn't respond the same way it should, the normal proteins would, to these regulatory pathways because you've lost that part of the protein. And so it goes to the nucleus. And when it gets to the nucleus of the cell, even though the cell is receiving signals that are supposed to be preventing it from doing this, it starts turning on a bunch of genes that are ultimately going to drive the formation of EHE, make the EHE cells survive longer, and make the EHE cells divide more often and more rapidly. And when they divide, they're going to pass this mutated form of TAZ-CAMP1 onto the daughter cell. So now you're going to get a tumor that has a whole bunch of cells that have this TAZ-CAMP1 oncogene that's not behaving appropriately, and it's too active. Also important for this is when TAZ-CAMP1 gets to the nucleus, it has to partner with another family of proteins called the TEADs in order to turn on these genes. And that will be important in a second and in other places throughout the talk. So just remember that in order to do these things, you need this partnership between TAZ-CAMP1 and other proteins called TEADs. So, okay, so if this is kind of, and we know that this TAZ-CAMP1 mediated gene expression is important to maintain the growth and survival of EHE cells. Okay, and so if we start thinking about how we could treat EHE, there's a couple ways we can do this. If we can prevent the cell from making enough of the TAZ-CAMP1 protein in the first place, or cause the cell to degrade it, and it's not there at all, then it can't go to the nucleus and turn on these genes. If we can somehow prevent it from shuttling to the nucleus, either by repressing pathways that are necessary to get it there, or by activating pathways that will restrain it or keep it into the cytoplasm and not let it get to the nucleus, that would also work. And then you can try to disrupt the interaction between TAZ and the TEAD protein so that they can't form that partnership that turns on these genes. And you may have heard about the TEAD inhibitors that are being used in clinical trials now to treat some EHE patients. That's what these drugs are trying to do. They're inhibiting TEADs and preventing them from interacting with TAZ-CAMP1 so they can't turn on the genes that ultimately are necessary for EHE survival and growth. Okay. Of course, we can also try to figure out which genes the TAZ-CAMP1 TEAD partnership are turning on that are actually causing these events, and try to block those with drugs or get rid of those in cells. And then finally, we can think about doing the same things that we do in a lot of other cancers, which is to just put things on the cells that kill the tumor cells, hopefully killing the tumor cells without killing the normal cells in the body. So, and this could happen independent of this pathway completely. It's just things that the cells need for survival and growth that we're blocking. Okay, so before I talk a little bit about the projects, let me give you a little background on how my lab got interested in EHE. It really all started with this guy named Guy Weinberg. Some of you may have heard of Guy. So he called me up in 2016 and he said, I want to organize a conference on YAP-TAZ and TEAD and targeting them in cancer, but I don't know anything about YAP-TAZ or TEAD. And so I need your help because you do know about YAP-TAZ and TEAD. My lab had been studying it for a number of years, and he was looking for someone to help him organize this. And he had kind of an ulterior motive to setting up this conference. And that was his son had been diagnosed with EHE. He's alive and well today and living with the disease. And so he recognized that there was not a lot of EHE research going on. And so he thought EHE is caused by the mutated forms of these two genes, YAP and TAZ. There's a lot of people who study YAP and TAZ, but those people aren't really interested in EHE or not focusing on EHE. So if we can get them to talk to EHE researchers, maybe they can share all their knowledge, help promote EHE work and give new ideas to researchers in EHE. And maybe we can also entice some of these folks to start looking at EHE. And so we hosted our conference. It was very successful. People were interested. So we had another conference in the next year in 2018. And it was at this conference that guy said, John, you should start studying EHE. And I said, I would love to start studying EHE. It's very interesting to me. I think that we could make some discoveries, but my lab is not funded to do that. And we can't just start using the money that we're currently being paid to research breast cancer to look at EHE. And he said, well, okay, write a grant. We'll give it to the EHE Foundation. And fortunately for me, the EHE Foundations formed a partnership, and they awarded my lab a seed grant that could get our work on EHE started. And around that same time, I connected with Brian Rubin, who probably all of you know, at both of these conferences, and he graciously agreed to start a collaboration. They shared some of the reagents they had that gave us the chance to get started. And Brian and I have been collaborating ever since that day on a lot of EHE work, some of which you'll see today. So it all started with this seed grant, and I'll kind of come back to why this is really important at the end. But let's talk a little bit about what we've actually been working on. And so I'll tell you about two different projects in the lab. This identifying regulators of Tascamta-1 is where we really initially got started with that seed grant. And then this drug repurposing is what we're up to now. And I'll show you some of the newer data that we have there. So as a Hippo YAP TAS biologist, I looked at the Tascamta-1 protein at that EHE meeting that I told you about, and I said, yeah, I can see why it's not really responding to the Hippo pathway to some extent because it's lost this one part of the protein, but there's a lot of parts of this protein that are still they're still able to be regulated by other pathways in the cells. And so we should be able to restrain Tascamta-1 if we can just activate the pathways that normally act on TAS in this region, right? And so that was kind of the idea. Let's come up with some ideas that we know will regulate TAS and see if we can get them to regulate Tascamta-1. And so we're really trying to find ways to do one of these three things that I mentioned, which we think would be a very effective way to treat EHE, to prevent it from getting to the nucleus, prevent it from interacting with TEADS, or to reduce its expression altogether by causing the cell to degrade it. And so this work really started with my graduate student, Ryan Kanai. And when we started this work, there were no EHE tumor cell lines. There were no mouse models of EHE. There was really no EHE that we could use to study EHE. And so we did what everybody else had been doing. And really at that time, everybody else was mostly Brian Rubin and Munir Tanas. And we took the Tascamta-1 gene, the blueprint for Tascamta-1, and we put it into fibroblasts. And when you put genes that cause cancer into fibroblasts, they kind of make the fibroblast behave like cancer. And so this is an assay called a foci formation assay. We basically take fibroblasts, we let them divide on a dish until they get really tightly packed together. And normal fibroblasts will stop dividing because they are receiving cues that tell them to not divide. Cancerous fibroblasts will start to form little tumors where they grow on top of each other. And you can kind of see these black dots are the little tumors. We call them foci. Only the fibroblasts where we put in the gene for Tascamta-1 make these tumors. And so we have a Tascamta-1 mediated tumor formation. And this gives us a way to then ask, if we do things to these cells and we can block this tumor formation, maybe that's because we're blocking Tascamta-1. It's a way to study the regulation or identify things that we can use to control Tascamta-1. And so this is done on a dish, cells on a dish. We can also take these cells and put them into mice. We can label them and stick them into mice, and they will form tumors, but the cells that don't have the Tascamta-1 won't form tumors. And so we can, again, use this to test things like drugs, or we can genetically manipulate these cells and ask whether those things that we do to the cells can block this Tascamta-1 dependent tumor growth that we see. And that's exactly what Ryan did in this collaborative study that we had with Brian Rubin's lab. Brian Rubin's lab had identified a signaling pathway downstream of Tascamta-1 that they thought was really important for the growth of the cells. And so we took the Tascamta-1 expressing fibroblast cells, and Ryan made tumors out of them in mice. Once the tumors reached a certain size, he treated half of the mice with a drug that blocks this pathway called trametinib. And then he left the other half not treated. The ones that he didn't treat, the tumors grew fast and got bigger. The ones he did treat with the drug stopped growing, and some of them even shrank. And so this demonstrates that we can use this model to identify drugs that repress the tumorigenic function of Tascamta-1. The problem with this is it's not fibroblasts, right? So we're sort of stuck using this fibroblast model and hoping that that's going to be effective in EHE. Nevertheless, Ryan's sort of pushed forward because, again, at this time, he didn't have any other models. And he developed one other assay. And this is an assay that measures the activity of tascampto-1 when it partners with TEADs. So remember, I told you that the functions of tascampto-1 that allow it to make EHE a tumor and allow EHE tumor cells to grow and divide are really dependent upon this partnership. And we can measure the activity or the amount of that partnership that's happening in the cells as a readout for how actively the tascampto-1 is turning on all those genes that drive tumor growth and tumor survival. And so this bottom part of the figure here is called a Western blot. It just measures the amount of a protein that the cells are making. And so when we force the cells to make more and more of this tascampto-1, and we use this reporter assay, we get an increase in the activity of that reporter. And that just tells us that as there's more tascampto-1, you have more tascampto-1 activity. And so it gives us a way now to put drugs on cells or to genetically alter these cells and ask whether we can repress the activity of this tascampto-1 TEAD partnership. And that will identify a pathway or a drug that might be effective in treating EHE. And that's kind of the approach that we took. Again, we went back to our knowledge as people who understand the Hippo pathway and the regulation of the normal TAS protein. We said, okay, there's a bunch of pathways that should be repressing TAS that should function on the part of TAS that's retained in tascampto-1. And so they should be able to repress tascampto-1 if we can turn them on in cells. And what Ryan did was he put in the genes that encode some of the proteins that would do this. They would repress TAS and ask whether they repress tascampto-1. And so this is, again, that reporter assay that measures tascampto-1 TEAD function. When you put tascampto-1 into fibroblasts, it turns on the reporter, as we expect. And when Ryan put in these different genes that should regulate the normal TAS protein, they do, in fact, block the activity of the tascampto-1 TEAD partnership. And so it brings it down to almost the level where the cells that don't have any tascampto-1 at all. And so this is really interesting because it says that we actually can regulate this protein. Up until this point, we really thought it was kind of an unregulatable form of TAS. And this tells us that we can get enough activity from some of these proteins in cells, we can actually turn off tascampto-1. The trick is getting the activity of these genes to go up in the cancer cells. And we were particularly excited about these two genes called AMP kinase alpha-1 and alpha-2. And the reason we were excited about these is because there are drugs that are already used in patients that can turn on these genes. So metformin and ICAR are two drugs that you can take that will activate AMP kinases. Sirolimus is a drug that you may have heard about because it's sometimes used in EHE patients. This drug acts on another part of a pathway that's sort of downstream of AMP kinase, but it has a similar effect to turning on AMP kinase. And so we thought, okay, there might be a way to actually turn on these genes and actually repress the activity of tascampto-1 in an EHE cell. But again, up until this point, we're using fibroblast cells. They're not EHE tumor cells. And so we're just sort of hoping that this regulation that we've identified will exist in an actual EHE tumor cell. Fortunately for us, this is about the same time that Brian's lab developed this transgenic mouse model of EHE. So these are mice that have been genetically engineered to express that same tascampto-1 fusion gene, and they get EHEs that are effectively indistinguishable from the human EHE disease. And so it's a good model to study EHEs. And we were able, or Brian's lab was able, with our help a little bit, to basically generate EHE cell lines from the tumors that form in these mice. And so we can now culture EHE cells on a dish and do all the same types of experiments that people do with breast cancer, melanoma, and in other cancer types to study the function of these tumor cells, to test drugs that we think might work on these tumor cells, to genetically alter these tumor cells to see if we can identify pathways that are important for growth and survival. Importantly, we found that we can use all the same tricks that we use in other cancers to manipulate these cells. So we can stably deliver genes to them. We can get rid of genes from them. And in this case, we've just expressed a protein that fluoresces under the microscope so that we can actually see the cells when they're in the mouse and when they're on the dish. But we can also alter pathways using this same approach. And so this allows us now to take EHE cells, manipulate pathways in them, like the pathways that we've identified here, and ask whether that can affect their ability to grow and survive. Importantly, we also know that these mouse EHE cells, like we predict would be the case in the human disease, are highly dependent upon this TASCAMTA1 TEAD partnership that I told you about. So if we take one of those TEAD inhibitors that disrupts the interaction between TASCAMTA1 and TEAD, as well as the normal YAP and TAS with TEADs, and we treat the cells with them at increasing doses. This is that same reporter assay I told you about that measures the TASCAMTA1 TEAD activity. The normal activity is set at 1, and then the drugs in a dose-dependent manner reduce the activity of this of TASCAMTA1. So they're doing what they should do. They're blocking TASCAMTA1's function by disrupting TEADs, and importantly, that also slows or decreases the viability of the cells on the dish. So if you take EHE cells and you put them on the dish and you let them divide over time, the control cells are they're gonna start dividing, and there'll be more and more cells on the dish over time. If you treat them with drugs that reduce their viability, you won't get that same increase in cells, either because some of the cells are dying or they're not dividing anymore. Either one of those things will be good for an EHE patient because we're either slowing the growth of the tumor or we're killing the tumor cells off, right? So this tells us that these tumors are dependent upon TASCAMTA1, which is what we want, and that TEAD inhibitors may work in these. But more importantly, it allows us to be able to measure drugs that block TASCAMTA1 activity, because when we block TASCAMTA1 activity, the cells they're not as viable on the dish. Right. And so now we can test those genes that Ryan identified to see whether drugs that activate them do the same thing. And that's exactly what he found. So if we take 2 different drugs that turn on that AMP kinase protein, we found that they're the green lines here. They reduce the growth rates of these EHE cells. So the cells don't divide over time. They may even die off a little bit. And that happens in both of the EHE cell lines that we've tested this in, as well as with 2 different drugs that do the same thing. And so this tells us that if we can somehow turn on this AMP kinase pathway in these EHE cells, it's slowing their growth rate on the dish, right? And so what we're moving to do now is to take those same EHE cells and put them in a mouse and get them to form a tumor, and then testing these same drugs in the tumor to see whether they can slow the tumor's growth rate or hopefully even kill the tumor cells off. And we call those preclinical mouse models of EHE, and I'll show you some of those models in a little bit later on in the talk. We also, of course, would like to make sure that these drugs work on human EHE cells. Everything I've shown you so far was done in the mouse EHE cells. And so once the human EHE cells become available, that this will be an experiment we would need to do. And then we'd like to know whether the way that the AMP kinase activators are working is by whether or not that's by inhibiting TASCAMTA1. So when we started the whole project, our goal was to identify things that repress TASCAMTA1, because that's a very specific way to target an EHE cell. And in that fibroblast model, that's what these AMP kinase activators are doing. But all we've shown so far in the EHE cells is that it kills this, or it slows the growth rate of the cells. And so we'd like to know if it's doing so by blocking TASCAMTA1 activity. And these are all experiments that Ryan's doing now as he's trying to wrap up his thesis work. Okay, and so now I'm going to tell you about some new data in the lab, a new project that is really looking at taking existing FDA-approved drugs that are already being used in patients for with other sicknesses or diseases, and repurposing them for use in EHE patients. And this is a really nice approach because it avoids the time that it takes to develop a new drug and do all the safety testing in the patients. They've already been tested in patients. They know that they're relatively safe. And so it's just a matter of proving that they will work in EHE, and then presumably the patients could go on the drug relatively quickly. So it's a quicker way to arrive at a therapy. And so what we're doing here is we're really looking for anything that will kill the EHE cells or slow them down from dividing, right? And so we're focusing on this step. They may be doing this step through this pathway, or they may be doing it independent of this pathway. It doesn't matter. We're casting a wider net. Anything that will kill EHE cells or slow down their growth is something that we're interested in looking for, especially if it's an existing FDA-approved drug. And so this is work done by a graduate student in my lab named Sarah. And so what she did was she took 190 different drugs that are already approved by the FDA to treat other diseases in humans. And she basically put them on the EHE cells and asked whether or not that they killed the EHE cells or reduced their viability. And so we have 2 different assays that we use to measure viability. One of them is called the viability assay. It just sort of estimates cell number, and then we directly count cells. So we put the drug on the cells, we let the cells divide on the dish for 4 days, and we count how many are left at the end of those 4 days. And so the untreated cells are going to increase in number. If we've reduced the growth rate or killed the EHE cells off, there'll be less cells in the drug-treated wells. And so we identified any drug that will reduce the number of cells left on the dish by at least 50% or more. And then we took those drugs and retested them in the second EHE tumor cell line, so that now we're looking for drugs that are working in two or more EHE cell lines. We also tested those same drugs in normal cells, because what we'd really like to find is drugs that kill EHE cells but don't kill normal cells, because if they don't kill normal cells, they're much less likely to have bad side effects in patients. Keep in mind that all these drugs are already used in patients, so whatever side effects they do have are at least tolerated, but we'd like to focus on drugs that are really specific to EHE. So this is kind of what the data looks like after Sarah screened all 190 drugs. And so what we would expect to see is that the vast majority of those drugs don't do anything to the EHE cells. And so we set the untreated cells at 1. So if the drug's not having much of an effect, it's going to be, its dots are going to be very close to 1. And that's what the vast majority of the drugs showed us, that they're not affecting the EHE cells' viability. But we identified this cluster of 24 drugs that pretty significantly reduced the viability of the EHE cells. And there were 24 of them. Sarah then rescreened that same 24 in a second cell line, as I told you. And this is what the data looked like. So we had a total of 24 drugs. 16 of them also affected the fibroblast cells, and so they're not specific to EHE, and we're not as interested in focusing on those, although they could still be good therapies, because again, these drugs are safe to use in humans. But we focused on the ones that were specific to EHE, meaning that they only killed the EHE cells and they didn't really affect the fibroblasts. So the gray bars stay high and the blue bars are low. Interesting for us was that 4 of these drugs are all in the same family of drugs, the statins. And so you may have heard of statins before. These are very commonly used drugs in patients. They control cholesterol. They're used to control cholesterol levels to reduce the risk of atherosclerosis and heart disease. And so 4 of the 8 drugs that we identified that are EHE specific were all statins. And so Sarah has been pursuing this pretty heavily. I'm going to show you some of her data. A medical student in my lab named Rosalind has been looking at some of the other drugs and focusing on those. And we don't have as much data there, but there are some promising results for some of these drugs as well. So we're not ignoring the other 24, but we're mostly focused on these statins. And so what Sarah did was she took EHE cells and she treated them with 3 different statins and did that same viability assay. And so the normal cells will grow fine. The really low doses of statins don't have as big effect, but as you get it to a little bit higher doses, it actually kills off the cells. And so the cell number actually decreases over time instead of increasing. And we can do these things called dose response curves that identify the dose of the drug that is very effective at killing at least 50% of the cells. And these doses are fairly reasonable and comparable to other studies that have used statins. And so that gave us some confidence that we're looking at a drug that might be usable to actually kill EHE cells. Sarah did the same exact thing in 3 more statins. So now we've tested a total of 6 different statins, and we've tested them in both EHE cell lines, and we get effectively the same results. So the 317 line is a little bit more resistant to the statins, but they're still effectively working on them. So the last thing that folks want to look at on a Saturday morning is these complicated metabolic pathways. So I'm not going to bore you with all the great details of them. But I do want to point out that the way that statins are thought to regulate cholesterol is by blocking this kind of crazy looking pathway here. And it's really the key part is this enzyme called HMG-CoA reductase. It's responsible for generating this thing called mevalonate. And so mevalonate is ultimately used to synthesize cholesterol by the cells. And so if a patient's got high cholesterol, they don't want their cells making more, so they give them statins, block the activity of this enzyme, and you reduce the amount of cholesterol cells are making. Mevalonate's also important for other things that cells need as well. And in particular, this thing is ultimately acted upon by other enzymes to modify proteins in the RAS GTPase and the Rho and RAC GTPase families. And these are very important proteins for a lot of cell functions. Including in cancer cells. And so when you treat a cell with statins, you're blocking all three of these pathways. To prevent atherosclerosis, you're trying to do this. But in cancer, we don't know which of these pathways is important for the cancer cells. And so that's one of the questions we wanted to ask. And so statins have been used in other cancers before, so this is not a crazy idea. There are some clinical trials now in some of the more common cancers that are looking promising. Interestingly, statins are also already known to act on YAPTAZ. And so the normal YAPTAZ proteins are regulated by the statins because they block this mevalonate, and that ultimately reduces the activity of this protein called Rho, which is really important for YAP and TAZ to go to the nucleus. And so it's pretty reasonable to predict that maybe they were also blocking EHE viability by doing the same thing to TAZ-CAMP1, blocking the activity of this Rho GTPase and preventing TAZ-CAMP1 from going to the nucleus, where it's going to partner with those TEADs and turn on those nasty genes that drive tumor growth and survival. And that's kind of what we were predicting. There was some preliminary evidence from DJ Pan's lab using a TAZ-CAMP1 expressing endothelial cell line where they showed that if they treated with statins, it did reduce the activity of TAZ-CAMP1. Again, this wasn't EHE, but it is supportive of our general idea that the statins are probably or hopefully regulating or inhibiting this TAZ-CAMP1, and that's why they're ultimately killing off the EHE cells. And so the first thing that Sarah wanted to do was just to make sure that we are actually focused on the right pathway. Statins are known to block these functions, but they can do other things to cells too. And so if these processes are what's important for EHE cells to survive, and that's why when we treat with statins the EHE cells die, then we should be able to rescue or prevent that cell death caused by statins by giving the cells back this mevalonate protein or this mevalonate product. And so if statins are blocking HMG-CoA reductase, and that's why the EHE cells are dying, if we just give back mevalonate, the cells don't need HMG-CoA reductase activity anymore, and they should do fine in the presence of statins. And that's exactly what we saw. So if we treat cells with statins, all the cells die. But if we treat cells with statins and give them mevalonate, they don't die. They survive just fine. And this happens at all higher doses of statins. It happens with all the statins that we've tested so far, and it happens in both the EHE cell lines. And so this tells us that the reason the statins are effective at killing EHE cells is because it's blocking one of these three pathways, either this pathway, or it's blocking cholesterol synthesis. And to kind of make a long story short, Sarah went on and kind of dissected this crazy pathway by either adding back some of these other components so she could rescue one part of the pathway without rescuing the other parts and ask whether that rescues EHE viability, or she can block some of these other enzymes and prevent one part of the pathway without affecting the other parts. And ultimately what she found was it wasn't the reduction in cholesterol that was what was killing the EHE cells. It was the combined reduction in the activity of these two families of proteins, the RAS GTPases and the Rho GTPases. Because if she restored these parts of the pathway, it restored viability. If she restored cholesterol, it didn't. And so you might be asking yourself, well, who cares, right? Statins are a drug. We can take them, it'll work. Sounds like, why do we care why they're working? And the short answer is it's always better to know how a drug is killing cancer cells. It also gives us other things that we might be able to target. So if the cells start to respond to statins, but then they stop responding, we might be able to go in and block some of these other enzymes and get the same effect and perhaps even reduce any side effects that might happen. So dissecting a pathway like this and figuring out the other parts of the cell's machinery that are necessary just gives us more ways to go after the cancer cells. And that's one of the reasons that we did this. And we do have some preliminary data that supports the idea that by blocking these pathways, we're really reducing TAZ-CAMP1 function. And so when we treat with statins, there's a lot less TAZ-CAMP1 in the nucleus of the cell. And remember, I told you that that's important for it to partner with the TEADs and turn on all the genes that make the EHE cells nasty. And so it seems like what we thought was happening is what's happening. But the bottom line is, what we really care about is, can we actually use statins to treat EHE, right? And so to do that, you got to do an experiment in a mouse model first. And so fortunately for us, those same EHE cell lines that I've been telling you about do form tumors when you inject them into mice. And so they will form somewhat slow-growing tumors that kind of mimic the indolent EHE, but they will ultimately grow larger, and the mice will succumb to the disease. And those tumors will metastasize or spread to the same types of organs that human EHE tends to spread to, the lungs and the liver. And we can also get them to grow directly in the lungs or the liver if we inject them into the mice. In this case, we put them intravenously. My lab's got another fancy technique called an ultrasound-guided intracardiac injection, where we take fluorescently labeled EHE cells and inject them into the heart, specifically the left ventricle. This allows them to spread throughout the whole body. And when we do that, the EHEs will form in the lungs, liver, and bone. And this is important because patients get EHEs in lungs, livers, and bones most predominantly. Those are the ones that are hardest to treat. And so we want to be able to test whether the drugs that we're interested in can treat all of these different EHEs that are forming in these different tissues just as effectively. Because if they only treat lung EHE but not liver EHE or not bone EHE, we need to know that in order to identify the patients that are most likely to benefit from the drugs. And so we have models now that will allow us to test these drugs, and that's kind of what we're working on now. Sarah's got some mice that are hopefully forming tumors, and she's going to start treating them with some of the statins very soon. We do this in kind of a fancy way. We take the EHE cells and we label them with two different things. One's a fluorescent protein that we can see under the microscope, so we can actually see the EHE tumors in the tissues that they're growing in at the end of the experiment. But we also put in another protein called luciferase, and this is the actual enzyme that's in the abdomen of a firefly that allows the firefly's abdomen to glow. And so if we put this into cancer cells, they will glow and emit light. And we can actually measure that light that's being emitted by the cancer cells in a living mouse. We just anesthetize the mouse, and we use a machine that we have that will measure the light emitted by the tumor cells. And so as the tumors grow bigger, more light will be emitted. If the tumors shrink after drug treatment, then less light will be emitted. And we can track the progression of the disease or the effect of the drug using this firefly enzyme. And then we can look at the end of the experiment to see whether there's less tumors and whether they're smaller. Right. And so what we're doing now is we're testing this in both an indolent form of EHE. And so when we put these cells in, there's a big period of time where the tumors that form don't really grow, just like is in the case in the human disease. And so we can ask, do the statins kill off that small tumor or prevent it from growing in the first place? Or we can wait until the disease starts to become more aggressive and the tumors grow more rapidly, and then we can treat the mice with statins and ask whether that prevents the growth or slows it down or prevents the EHE from spreading to other places. And then we can use this intracardiac model I told you to ask whether the statins, if they are effective, are having the same effect no matter where the EHE cells are growing, or whether they're only effective in one tissue but not the other. Again, important to know when we start thinking about this from a treatment perspective. Okay, so I just kind of told you that what we're up to now is to take and test the statins in mice. Again, we haven't done this in human cells yet because we don't have human EHE cells to do it in. Once those become available, the foundation is kind of working on developing those. Once they're available, we will certainly test them in that and in the EHE human lines. We're also looking at ways to partner with clinicians to try to figure out if we can do a retrospective study. And so this is basically looking at past EHE patients and seeing if there are any who were on statins and then comparing how well they did to patients who are not on statins. And this would just be because those patients were on statins for other reasons. And so if we can see that in general, the statin-treated patients do better, then that gives us some evidence that everything we've learned so far in a mouse is maybe relevant to some patients, and that's very good news. If these things look good, which is what we're hoping, then the next step would be to start partnering with actual clinicians and oncologists to try to move forward and think about ways that we can actually start testing statins in EHE patients. And then independent of this, we're also kind of using the same approaches that we use to identify the statins to screen a much larger library of drugs. We're doing it in a slightly different way, but hopefully we'll have some other candidates there in case the statins don't prove to be as promising as we're hoping they are. And we're also looking for biomarkers that can be used to measure how effective drugs like the statins are in the EHE patients, as well as monitor their disease progression. So if you're a patient that's in active surveillance, it would be nice to not have to wait until you do a scan to know if your disease is progressing. If we could just take a simple blood draw and say, oh, there's more of this EHE marker in the blood, we should start treatment right away. That would be good. And then also with respect to treatment response, there are some drugs that have been effective in EHE, but they don't work in every patient. And so you'd like to not waste the time of trying to treat somebody with a drug that's not going to work. If you have a very quick readout for the fact that the drug is working, then that allows us to take patients who are not responding and put them on another drug, or keep the patients who are responding on that drug that's working. And so that's one of the sort of longer term goals of the lab, is to try to find biomarkers that will allow us to do that. Okay, so that's most of the data, and I'm happy to answer questions. Before I do that, though, I just want to mention a couple things. The first one is everything that I've told you about and everything that we need to do to take what my lab has done so far and actually translate it into something that's going to help patients are things that the EHE Foundation is actively doing, and they're playing a very important role in this. And so EHE is a rare disease. There are not a lot of ways for us to fund that research from the federal government and other sources. So the EHE Foundation is really responsible for a very high percentage of all the funding that is going to labs like mine to do EHE research. They obviously host these really great patient advocacy and outreach events like this one and others that they do, and they connect with patients and connect patients with clinicians. And so that's a really important function that they serve. I mentioned that we may need to start looking at human specimens and human patients and human patient data. We need human cell lines to test these things, and the EHE Foundation is doing all of these things. And I'll show you in a minute the links to the biobank and the patient registry that you can exploit to share your information. Collecting this data and having it available to researchers like me is a really important step in understanding this disease and predicting how well some of the things we're learning in cells and mice are going to work in humans. Okay, and so the foundation is playing a really critical role in generating this data for us to use. And then they really help people like me connect with other EHE researchers and clinicians. And so as I'm going to show you, we interact a lot with other EHE researchers, and that's a big part of that is that the foundation is very good at bringing us together. And they're very good at connecting us with the doctors and the oncologists that are going to help us understand the human disease even better and start to collect some of this information. And ultimately, hopefully, if we get to a point where these drugs look very promising, to set up these clinical trials and other studies that we would hope ultimately help EHE patients, right? And so the foundation is playing a very critical role, and that kind of brings me to your role. So as patients and advocates, you have a lot of power. Your donations and your voice are what helps the EHE Foundation do all the things that it does. And I think, as I've hopefully shown you, a little investment in a lab that would not otherwise be able to do any EHE research can have a big impact. And so to show you the rest of the timeline, after we got that grant, that allowed us to generate preliminary data that led us to other grants. And I'm in the process of submitting a grant now to the federal government for hopefully a larger sum of money that will allow us to keep doing EHE research. And so a small investment on the part of the foundation has allowed my lab to grow and continue to do EHE research. We've been involved in some of the studies, as Denise mentioned, that have, in collaboration with Brian Rubin's lab, that have made some advances in EHE, and hopefully we'll be publishing some of the stuff I've shown you as well soon. So we've made some achievements, again, with this small investment. It's allowed my team to grow from just one poor graduate student who's doing all the work to now several graduate students and technicians. And I think it's also kept me engaged in the EHE community. It's very hard for us as researchers to fund our labs in general, and we have to spend a lot of time and energy sort of chasing that funding. And even if you're in love with a disease or a area of research, if you can't get that funded, you have to kind of change course and focus on things you can get funded. And so by remaining funded in EHE, I can stick with something that I've become very passionate about and be able to continue researching it. The foundation has also been important in sort of advocacy and keeping me as an advocate as well. And so I mentioned Guy and I organized this conference where we're trying to bring EHE researchers and TAS researchers together. And so we've just had the 8th annual one, and they've all been very successful at really raising awareness of EHE in the YAPTAZ world. YAPTAZ now has EHE sessions at all their major meetings. Most of the YAPTAZ researchers are now aware of this disease and at least thinking about it on a regular basis. And so, and those researchers have helped us EHE researchers learn a lot as well. And just to tell you about it for another minute, again, the goal is to connect YAPTAZ Hippo researchers, of which there are thousands, to EHE researchers, of which there are not that many. Help us establish new collaborations, maybe recruit some new EHE researchers. And I think we've done a good job with this. Most of the grant applications that the EHE Foundation got this year have come from people who have attended this workshop. And so we've enticed them into the EHE world, and now they're applying for grants and hoping to start their own projects. It also allows us EHE folks to kind of get together and talk a lot and share ideas. Rather than competing with each other, we're working together. And so the way that this works is we have these science sessions in the morning where we present our work and talk science. And then we go out for these walks in the woods, which, because it's in Telluride, Colorado, a very beautiful ski resort area, they're very nice walks in the woods. And the whole time we're walking in the woods, we're talking science. And so at the last meeting, there were 4 EHE labs represented: my own, Brian Rubens, Munir Tanas, and Sandra Pasquale. And the 4 of us all hung out a lot. We went on this long hike to the top of this mountain up here, and we just talked about EHE and the ways that we could work together the entire time, and we weren't gasping for air in the 12,000-foot elevation. But it was a great time to kind of meet everybody, and we've continued to work together since then. So that's the goal of this workshop. The EHE Foundation has supported the travel of some of these EHE researchers to that workshop, and they've helped keep us engaged in that as well. So they play a big role in that. And so that brings me to 2 more things that you can do. The first is that EHE patient registry, where you can enter your information, tell us a little bit about the disease. And the other one is the biobank. If you're having a procedure, you can donate your tissue, and researchers like me can use that to help advance what we know about EHE. And I believe that the links to those are going up in the chat at the moment. And the other important thing to keep in mind is, as patients, your voice is important too. As cancer researchers, we are almost entirely trained to focus on curing diseases or understanding the disease so that we can cure the disease. Clinicians are focused on treating patients, but neither of us are really always aware of some of the other challenges that patients who are living with the disease face. And so, yes, we want to cure the disease, and that's what you want us to do too, but there may be other things that you're dealing with as a patient on a day-to-day basis that we can help with. We just don't realize that they're happening. And so by letting us know what challenges you face, what problems you face, what questions you have about life decisions that may affect or be affected by your disease, this can open up new ideas of research for labs like mine. And while we're working on the cure, we could also be working on things that might improve life for people who are living with a disease. And you can also offer us information, and that's part of what this patient registry is. So there are things that only a patient knows that we can't know by looking at under the microscope, or doctors can't know by looking at you, that can be helpful. There are useful pieces of information that we didn't realize. And so sharing that type of things in the EHE community and with the EHE Foundation is an important thing that you can do. And with that, I'll stop and thank the members of my lab who, this picture is not up to date, doesn't have everybody in it, but Ryan and Sarah are the main drivers of the work. Chloe was a technician who helped out a lot, as well as Kayla. Emily works on other projects in my lab. All this work has been funded in large part by the EHE Foundation, also the Cleveland Clinic, and the Robert E. Peterson Foundation. We did get a grant from the DoD in collaboration with Brian, so we did get some outside funding, and we're working on more, and hopefully that will come soon. And I'm happy to take questions. Stop sharing. Okay. So Denise, I don't know if you've been kind of monitoring.
I am here. I apologize. I was trying to click into the chat, and I clicked on something else on my computer. Thank you so much. That was amazing. Absolutely amazing. I think everybody is inspired and you got a great science lesson this morning, whether you wanted one or not. If you didn't think you were coming for science, you got great science.
I promise I did not make up all the names of those enzymes and stuff. They're real things, but
absolutely amazing. Really, really great. Okay, let's jump to some questions that came in early on. I'm just switching over to my other screen here. So Fiona wrote early on in your presentation, does EHE eventually convert to the aggressive form, or can some of us just die with EHE and not necessarily from the disease?
I think a lot of EHEs convert to the aggressive form, but not all of them have to. Obviously, it depends on the age of the patient as well. We'd like to see people dying of old age and EHE, but not because of EHE. But I think as most patients recognize, waiting around to see whether you're one of those people is not ideal, and that's why we're also trying to treat the indolent disease. So the answer is no, it doesn't always convert. We don't really fully understand why it converts. There's some evidence that there's another mutation to a tumor suppressor gene that can drive that conversion. But there are certainly patients that don't have that mutation that convert, and there are certainly ones that do have it that don't convert. So it's a little bit probably a combination of things, and that's one of the things that having a lot more patient information will help us understand that. These are the types of things that are very easy to do in other cancers because you have thousands of patients and you can look at all of the circumstances and make better predictions. It's much harder to do that with an ultra rare cancer. And so the more patient information we have, and we as the collective EHE community, not my lab specifically, is good for that.
Yeah, great. Someone also asked, does EHE share the gene mutation with any other types of common cancers?
Right. So that's a really interesting question. And one of the things that makes, and I apologize in advance, EHE fascinates me as a scientist. And it's weird to say a disease that some people are afflicted by is fascinating, but you want people to be fascinated because we as scientists try to solve problems. A fascinating thing about EHE is that you never see these translocation events in any other cancer, even though we know that YAP and TAZ activation or mutations that cause YAP and TAZ to be more active are common in a lot of cancers. You don't see the specific translocation event in EHE, and you really don't see any EHEs that don't have one of those translocation events or something similar to them. So it's very specific to EHE, and that's a good thing because it tells us that that's probably the real key to curing the disease. The only way you get an EHE is with this translocation, and so the EHE is addicted to the translocation.
And the translocation, just to clarify for everybody, is that TAZ-CAMTA1, is that oncogenic fusion,
right,
or the YAP TFE3.
Correct.
And then Milda asks, what characteristics does EHE share with other cancers?
So almost all cancers have similar characteristics in the sense that they grow when they're not supposed to, the cells divide when they're not supposed to, the cells survive longer than they're supposed to. They have to be able to recruit a blood supply when they grow as a tumor. They tend to have mutations that accumulate. EHE doesn't accumulate as many mutations as most other cancers do. It's a quiet tumor that way, so that's a little bit different. It spreads like a lot of other cancers do. That spread itself makes patients harder to treat, but unlike other cancers, that spread does not necessarily mean that your disease is very aggressive, right? So when we're looking at breast cancer and melanoma, the presence of metastasis is a very late stage event, and that tends to be a very bad sign for patients. It's very predictive of an aggressive disease that's going to be very hard to deal with. In EHE, you can have metastases that are indolent and not growing and behaving just like the primary indolent tumor. And so while metastasis isn't good, it is not as dire as it is in other cancers. And so that's kind of a unique feature of EHE. Other than that, I think, this cancer is very similar to a lot of other cancers. There are differences on a molecular level. Obviously, they have a weird microenvironment of their tumor that makes them very characteristic. It allows pathologists like Brian Rubin to identify them very easily by looking at them under the microscope. We think that's interesting and important as well. But yes, it shares a lot of common characteristics with other cancers.
Mm-hmm. Your comment about the metastatic disease, widespread metastatic disease is good because We know that many people are diagnosed with EHE and they have metastatic disease at diagnosis. It's incidentally, discovered. But having like stage 4, if you go by staging, right, of what breast cancer and other cancers are staged at, is not really aligned with EHE in general. So it's a good call out for particularly for people who are newly diagnosed. Maureen writes, can EHE patients switch from one TEAD inhibitor drug clinical trial to another TEAD inhibitor drug clinical trial without disrupting data of the new TEAD inhibitor drug trial? So I don't know if you can answer this one, but you know the science of the TEADs.
So I'm not a clinical trial person, and so you'd have to ask the coordinator of the clinical trial. That my guess is they don't want patients switching. My suspicion is that the reason you're asking this question is because we have a discontinuation of one TEAD inhibitor trial, and in that case, you would probably be enrolled as a new patient, but I don't know that for a fact. So I think that's a question you'd have to discuss with your oncologist. The science behind this is a little bit different. These TEAD inhibitors are not all doing the same thing, and it's something that we in the YAP-TAZ TEAD world are really trying to sort out. There's such promising drugs that everybody's been rushing to kind of get them out there and start working on them. But then us biologists are kind of behind a little bit in understanding how they're working and what their functions are. And there are 4 TEAD proteins. Not all the inhibitors block the same proteins with the same efficacy. And we don't yet know whether individual TEADs are always working in EHE or whether they can kind of switch TEADs. And so it is a little bit complicated, and my guess is one inhibitor is not going to be the same as the other, and we don't yet know which one to try. But I think if they do work, they are the most obvious way to treat this disease that I can see right now, because it should directly block what we are hoping to block.
Good point there, that these TEAD inhibitors are different, and please definitely check in with your doctor regarding trial. Fiona asked a really good question. I'm glad she put this question in. Is this evidence enough to ask oncologists to prescribe statins to EHE patients?
Right. So I will echo Maggie's disclaimer that I am not an MD doctor. I'm a PhD doctor. I am not trained to treat patients. I've never seen patients. I did not go to med school. I teach in med school, but I didn't go to med school. And so I'm not giving any medical advice at all. So everything I'm going to tell you is just my opinion. You should absolutely talk to your doctor before you make any decisions about your treatment or things you're going to take. These drugs can interact. All drugs can interact with other drugs and be affected by things you're already on. And so you don't want to make decisions without at least consulting your doctor. If you don't like what your doctor tells you, then consult a second doctor. And the only piece of medical advice I will give you is if you're not seeing an oncologist that understands or specializes in EHE, you should try to contact one and get a second opinion from them or get seen by them, because even the greatest oncologists on earth have not necessarily treated EHE, and they won't have the same experience treating this specific disease. And so it's really a good idea to talk to the foundation, have them help you identify a clinician that sees a lot of EHE patients. Mm-hmm. So to answer your question, I'm not going to tell you to go out and ask your doctor to put you on statins. Is this evidence enough for any other drug or any other trial? It would not be enough. We need to look at the mouse models. We need to get some data in human cells before I think we would think about prescribing it to patients. That said, statins are very somewhat benign drugs, right? People go on them for a lot of reasons. Some people just put themselves on statins because they're thought to be this wonder drug to prevent a lot of diseases. And so having a conversation with your doctor about whether statins would be reasonable for you is never a bad idea. And but I would be surprised if your doctor's like, oh, you saw a presentation, yeah, sure, you can go on statins. And I think we need some more data. But it is a relatively safe drug. And a lot of people go on it just because they want to control their cholesterol. And so if you have high cholesterol, then going on statins might not be a bad idea for this reason as well. Something you can talk to your doctor too about.
And your point, you pointed to patients' contribution of other ways, like to the registry, et cetera. I mean, it is important in the other projects that we're doing, around the world. There are clinical registries, we have patient registry. We're gathering medical records and data from other from other sources, but all of that data collectively builds the case. And we need more of that data because having 10 patients here or 5 patients there won't be enough. So donate your data is the moral of that story. Sandy writes, if I'm already taking, and I'm going to butcher this word, rosuvastatin for cholesterol reduction, should I consider changing to simvastatin or one of the others?
Right. So we haven't really dissected the different statins closely. In our hands, every one we've tried has worked. And when we're doing experiments in a dish, that's very different than a mouse. Some of these statins have longer half-lives in humans and in mice. And if it's an approved drug for cholesterol reduction, it's probably doing a very similar thing to the other statins. And so I wouldn't at this stage predict one's going to work better than the other. And if your doctor feels that that's a good statin for you, then it's probably fine. You can certainly ask that question. We're looking at doses that are a little bit higher than the doses that people typically take for cholesterol reduction. And in the other cancer clinical trials are also using slightly higher doses. But for the most part, the statins all seem to work in a very similar way, and they have just slightly different behaviors in the body. So.
Okay. Thank you. Sharon writes, will the statins possibly work together with sirolimus, or are they independent of each other?
Right. So it really depends on how they're working. Our data kind of suggests that they're not only just blocking TASCAMP1, they're also doing other things. That's probably the same. The same is probably true of sirolimus. So, one of the things we always think about is what drugs would work in combination that better than they do alone, and whether or not they would be bad to take in combination. And those are the types of questions that we can start also asking using the models we have. So we can start testing the statins with the TEAD inhibitors, with other drugs like sirolimus, and kind of see what they're doing. Obviously, again, your doctor is going to better know what the side effects are. Some drugs you don't want to take together because they have added toxicities and things like that. So it's a question you should ask your doctor before you take statins if you're taking sirolimus. And sometimes drugs will reduce the effectiveness of other drugs too. And that's another thing you have to be worried about. And your clinician is going to be trained to kind of understand that, have databases that will tell them not to put these two drugs together better than I will. All we can do is ask in cells, do they work better together than they do separately? The sirolimus works well in our models as well. And there's some evidence already being published from another group that sirolimus is working in some EHE models there. So testing them together would be a great idea. We just haven't done that yet.
Great. Okay, Hugh, Leonard, Hugh, John. Hugh's on, and he's asked a couple of questions, so I'm going to throw all of these at you. John showed different breakpoints for both TAS and CAMP1. This suggests that TAS CAMP1 could have different forms depending on which breakpoints are involved. Is this the case? And if so, could these drive variation in disease presentation and response? He also asks about whether or not you've looked at sirolimus and run similar tests to statins. And are you looking at GDF15 as a biomarker? So,
right. So first thing I should say is Hugh played a huge role in that first grant that the foundations collectively provide to us. So you can all thank Hugh for my presence here, and among others, but Hugh was a major part of that. And so thank you, Hugh, for supporting us and getting me started. I sort of answered the sirolimus question. We have tested it at a sort of superficial level, more as a control, because we sort of already were aware of Sylvia's work on sirolimus that is coming out of the Italian lab there. And so it does block viability. It's very potent, actually, in blocking viability of these mouse EHE cell lines at higher doses. We haven't tested it in parallel yet, and we haven't tested it in the mouse models yet. We are using GDF15 mostly as a transcriptional marker yet. We haven't tested it as a biomarker in the mouse models, but again, the Italian group has demonstrated it's a decent biomarker. So it's an obvious one to look at when we start treating the mice to see if the levels of it go down when the tumors go down. And that's something that we're going to be doing. We're designing an experiment to look at lots of biomarkers at the same time. And I would predict that one will come out. And so we'll kind of know that information when we do that experiment. The breakpoint question is a very good one. So this is actually really important, and some of the research we did early on was really focused on this. So that where the breakpoint falls, some patients have part of the TAS protein called the WW domain, and this is just an organization of the protein, but it's an important organization of the TAS protein because lots of things that regulate TAS bind to that region of the protein. And so if a patient has that domain, they may be susceptible to some treatments that another patient that does that's fusion doesn't have that domain would be refractory to, because the tascamta is going to ignore any regulation through that domain. So we try to test them both. We have a version of tascamta that has an intact WW domain and another version that doesn't. And so far with all the things we've looked at, now we don't have that in the EHE cells because the one that was designed is all the same. But in the fibroblast model, we don't see any differences in the two different forms of tascamta with the AMP kinase inhibitors or any of the other genes that we've tested. But it is possible that the breakpoint might influence some of the regulation, definitely. As far as disease presentation, that's hard to know, but that's also possible. So what we don't really know is whether a lot more tascamta activity makes the disease more aggressive or not. It's an interesting question that I think we need to sort out. There is some evidence that if you have too much tascamta TEAD function, that's actually bad for the cells. We've done some things to our cells that increase the activity of TAS and it kills the cells. So there might be kind of a sweet spot of activity that's not too low, not too high. And if you get any higher, that's bad for the cells. If you get any lower, the disease doesn't progress as fastly. And that can certainly be influenced by the breakpoints depending on what other pathways are regulating it. So I think these are all really interesting questions that we can start thinking about.
Mm-hmm. Yeah. I don't think I've missed any questions. You get lots of thanks from the crowd and so much hope. Lots of thanks on sharing a point of hope, really. I wanted to do a call out to you. You mentioned the biobank. One of the things that's really important, you have models. We are in the process of in the hopefully final stages of having human cell lines available. But even with the models we have, it's still important to have more models. So if people are in any situation having fluid drains or a surgical procedure, a tumor resected or a transplant, anything, biopsy is not really our ideal tissue to collect, but a needle biopsy, that is. But if you have a full tumor resected, we would want that. But getting that tissue to labs like John is really important to develop more models, because the more models we have, then we can have better preclinical data for all of these drugs that we're trying to test.
Yeah, that's a really important point. I mean, other cancers that we study, we have hundreds and hundreds of cell lines that we can work with, and even that makes it hard to take what we've learned in cells and effectively treat the human disease. And so the more we can have different models, the better we're going to understand the disease. And that's definitely the case. We're really happy to have the ones we have. It was many years before we had those. So it's a huge advance, but the more the merrier for sure.
Yeah. Yeah. Okay. I don't think that I have anything else. As Maggie mentioned, we will publish a transcript along with the recording of this on our YouTube channel. It'll probably be posted next week. You'll see it. Maggie will share that in our news. If you have any questions, feel free to always email us, and we can toss those over to Dr. Lamar. You'll hear from him again next year, I'm sure, when we're having our conferences and meetings, and we'll share updates from his research as they come available. So thank you very much.
Thank you.
Thank you. Thanks everybody for coming today. Thanks for giving your time on a Saturday. John, thank you for coming and taking Saturday too for the community. We really appreciate it. So glad to have you. Alrighty. Be well, everyone. Take care. Bye-bye.